Everything over.
Okay, let me just sort of go back a little bit then. Um, so I was generally giving some, you're raising your hand again. Okay, so okay, so the what we'll be using today is a software called Salmon that circumvents this really time-consuming step and that in star that generates these aligned reads. So we're not going to be generating Salmon BAM files, these sequence alignment maps that we did using star, but instead we're going to be sort of mapping our reads to a reference transcriptome. So this reference transcriptome is up here, and these sort of T1, T2, T3, these are all of the exons in the transcriptome. Essentially, the way that Salmon works is that each read comes in one by one, and we can sort of build out a table for each read where it may be suitably aligned, suitably aligned to, which reconnected again.
Yeah, you were disconnected again.
Oh, 1 second. Let me reconnect
See,
and disconnect.
yeah, let's give it a second. Just pause recording for now just to figure this out. No, it's everybody. I had to do my.
Okay. So just what was the last thing that sort of everyone heard? Hello?
I think essentially like salmon to sort of go past all these like really long checkpoints that are done in the other program.
Okay, yeah. So yeah, so I guess I can go back to give a little bit more intuition on what the algorithm is doing here. So we have a reference. So instead of mapping to a reference genome and having annotations, having these two different files, Salmon uses just a reference transcriptome. So the idea is that we have the sequences for different cDNA fragments that are or have been mapped. These are in like this T1, T2, T3. These are approximately like the exons that we know about. And a read comes in, and we sort of get an idea of where the beginning and end of different patterns match, and we can group reads into these sort of relationships with one another that sort of tell us which one of these sort of exons each one of the group of reads map to, and that sort of grouping gives us a probabilistic estimate of um, how many transcripts we have for each isoform of a gene, and then accordingly, what the sort of gene counts should be here under different kinds of normalization parameters. So part of the reason it takes faster, it's a little bit faster, is because we're using a smaller reference. And it's also this underlying probabilistic estimation gives us ways to model different kinds of biases that might exist, things like this bias in GC content. And we can also go back and give us some level of understanding of the variational noise that exists when we quantify gene expression. Any questions about this? Any specific questions about this algorithm? Okay, so let's. Oh, there's a chat question. No, okay. All right. So let's now. We're going to switch now to the exercises again, and what we'll do first is quantify the reads, and then we'll uh, come back to we'll come back to the slides when we import import our data. So what I've done, I've opened up another terminal shell here, and I'm gonna again ask for a 3-hour interactive shell with, let's just say, 8 GB of data or of memory. And we've already downloaded Salmon. It should be in our software folder. So let's take a look at that. It's in our CD folder. Oh, we're CDing into our base folder here and then changing our directory to software. And as we did on Tuesday, the Salmon executable is in this folder, but it's sort of embedded in the bin directory of that subfolder. So if we have to run Salmon, the executable is in this massive path /salmon. So the goal here today is to sort of put all the sort of concepts we've learned about scripting in Bash all together so that we can process all 4 of these FASTQ files in one go. So let's start with this first chunk of code here. So the first chunk of code here, there we go. First chunk of code here, I'm copying it back into a text editor so that's easier to run. I'm gonna change the directory to my data directory. Actually, let's change to the reference directory first. Let's change our directory to the reference, and we have to go grab a
People like us in the middle states have to ride it out. All these southwestern states might have a chance, but New York will have title develop all of
whoever is
It's a lawyer.
Yeah, Eloy, can you so we're gonna so now we're gonna have to grab our Eloy. We have to grab our hey Eloy.
If you're the host, are you able to mute him?
Yeah, I should be.
States have to ride it out. All these southwestern states might have a chance, but New York will have title and develop all of the Northeast.
There we go. Okay, so we have to grab our reference first. So the reference here is going to be the reference cDNAs that we got from Ensemble. So we're gonna go back to that FTP site that we looked through on yesterday. So if we go to just this part of it, right, the ftp.ensemble.org/pub, that's going to get us to all of the different releases that are available to us. And here, the most recent release is 109, like we've been using so far. So this right here is going to be release 109. And now we're looking for FASTA files because we're looking for assemblies of sequences. We're going to go for Homo sapiens. And now instead of DNA like we use for star, we need the cDNA reference. So the full link address here that we'll need is what's on the screen here. So this FTP site release 109, but the rest of it is exactly the same because we're still on the same genome build, this genome build of GRCH38. So I'm going to download this into my reference folder, and it'll take a little bit of time. So once we get it downloaded, we'll unzip it and take a look at what this file is and sort of compare it to the DNA assembly that we downloaded. So any sort of questions on this? Any trouble people are having with this?
Still making the code.
Okay.
One moment. I got it downloading right now.
Great. Anyone else having trouble with this?
No.
Okay. All right. So as we wait, just to sort of give a little bit of context to what we're doing at first here is the same thing we did for STAR was to take our reference genome build, that chromosome 18 FASTA file. We took that file and the reference annotations in our GTF, and we created what's called an index for index database for STAR. We're doing a similar thing for Salmon. We're building a transcriptome index using just the cDNA reference assembly that we grabbed from Ensemble here. So that's the first step here. All right. So now I'm going to unzip my cDNA FASTA. And let's print out the header, first 10 lines of this cDNA FASTA file. So what you can see here is it looks a lot different from the FASTA file that we downloaded for chromosome 18. Since this is cDNA, these DNA sequences correspond to exons of different genes. So what we see here first is this is now a tab sort of delimited text file where the first line is a header for a specific transcript here. We can see it's an ENST ID. The T stands for transcript. Yesterday we looked at ENSG IDs. These are ensemble IDs for transcripts for T and genes for G. And it's right here it says that this is a cDNA. It tells us what the chromosome here is. Gives us the start and stop positions. This chromosome here is like a scaffold, so it's not really a recognizable chromosome name here. Then it tells you what gene that transcript belongs to, the biotype of the gene in the transcript, the symbol of the gene, a description. And then the next line here is just the small sequence of that cDNA fragment. Similarly, in the next line, we see another ENST ID for a location in the chromosome on chromosome 14. There's the gene coordinates here, the gene it belongs to, and all the associated information for that gene, and then another sequence. So as you can see, these cDNA fragments as references, these are really short locations. So the mapping is that we can actually run into a sort of statistical challenge of each read from our raw data aligning to several one of the cDNA fragments. The goal of the
I have a question. For this cDNA library is for all genome or just for chromosome 18?
This is the full genome. So
Full
everything we do today is the full FASTA file, full genome, and this will sort of give you the main reason to use Salmon over Star, which is computational speed.
Mm-hmm.
Okay, so this is giving you so as I mentioned, this sort of this annotation file here, this FASTA file, is for any annotated exonic region that's been reported and cataloged by Ensemble. So if we take a look at just how many lines this file has, we have 6.7 million of these. So divide by 2, we have 3.4 approximately million cDNA fragments. The sort of catch here, however, is that these are the human transcriptome, or even for other mammalian species, these aren't sort of one size fits all annotations. Different cells, different tissues even express different exons and transcripts at different time points during development of the tissue, etc. So this is only giving you a snapshot of the transcriptome, which sort of gives us a disadvantage of a method that is dependent entirely on these cDNA annotations. So this goes back to a question that we talked about yesterday, where what if we don't have a good reference? Can we do de novo assembly of a transcriptome? The answer there is yes, and oftentimes there's sort of a reason to run a de novo assembly of a transcriptome, but essentially having the map of the reference genome really helps in that setting. So say you're interested in a specific gene that you believe to have a bunch of transcripts that are specifically expressed, say, in, I don't know, like differentiation of the cell, of a sort of neuronal cell or something. You can run RNA sequencing, either short read or long read, and use these de novo transcript assembly methods to build up a novel transcriptome that's specific to this sort of period of time, and from there you can annotate new exons or transcripts that are specific to this cellular context that you're interested in. So this is sort of the push and pull of RNA sequencing here. You have to be very careful and very deliberate with what the goal is for your experiment, and accordingly choose a pipeline. So the sort of methods we used yesterday for STAR actually gets us to aligned reads to the genome. We can still use those aligned reads and process it in another way to sort of identify exons that aren't annotated in our GTF file. At the same time, we can use a small subset of samples to build up a transcriptome assembly, update a GTF file, or update our cDNA library here, and then use this library to sequence short read RNA sequencing or quantify RNA short read RNA sequencing data using Salmon for a larger number of samples. So does that sort of make sense? This compare and contrast of references.
Yep.
Great. Okay, so there's a question in the chat. So is this so Jejun asks, so is this reference file the collection of all the known isoforms so far? Yes. So this is Ensemble's catalog of known isoforms of genes. And I think a lot of the long read RNA sequencing literature cites that this is probably around 20 to 50% across different cellular contexts. So we still are probably in the range of 60 to 70% missing in terms of transcripts. Not all of these transcripts are going to code for proteins. Not all of these transcripts even have regulatory functions, but they're transcripts nonetheless. And there's a lot of work left to be done in annotating different kinds of cells, different kinds of tissues. How often does it update, and will it change the name with updates? Yes, that's why we specifically looked for release 109 instead of release 95. So if you look at the Ensemble browser here, if you go back to pub, if you go back to this, you can see that release 109 was updated today. But then release 108, the last time it was updated was in October, same with release 107. So it changes and the names change, and accordingly, the same thing with GenCode. The GenCode is another way to find these annotations for different exons and transcripts. What these also, you can get legacy, you can get legacy builds here through this release history and Schroll releases, and this will tell you the GenCode releases from 43, 42, all the way back to the first releases. Does that answer your question? Great. Okay, so now what we're going to do is generate a reference index transcriptome. And the first step here is I'm going to make sure that I have my path. Oops, that's an issue. I'm going to make sure that I have the proper path to my Salmon build. So my Salmon build is contained in this giant path. What I'm going to do is save it as a variable name. So now if I go back into my reference folder, I can call the Salmon help by I can call Salmon itself by typing in $Salmon, and that's going to call up the Salmon program. Okay. So now what I'm going to do is run an indexing command here. So this first bit here calls the Salmon program. This tells us that this is the index command. The -t tells us that this is the cDNA FASTA library that we should be using. This I here is sort of the name that's given to the folder that's going to contain all of the files for this reference transcriptome. And this -k here, I'll talk about a little bit but let's get this running first, because it'll take a little bit of time. So everyone, try to give me a thumbs up or something to just a thumbs up or something just to or just stop me if you are getting errors or something, but this should be pretty straightforward in terms of so I'll give you, I think, 2 minutes just to, if there's any errors, and if not, and then we'll start. We can talk about what this K parameter here is.
Arjun, sorry.
Yeah.
How do you? Okay, I just wanted to know what the command was before it, like the whole pathway. So we're going from our. I'm currently in QCB W5a, so should I be in reference right now before I write that out?
It doesn't matter when you write that out. You can always rerun the entire thing, right? It's just basically make sure
Okay.
you know where your Salmon folder is and where inside of that Salmon folder is the bin subdirectory, and inside the bin is your Salmon executable.
Okay, gotcha.
Okay, so what this K parameter here is, it's this hash table that I talked about where they were chopping up the sort of cDNA into these smaller chunks and trying to match up or map up the reads with the cDNA library. It's looking for a sort of sequence length that it's going to look at. That's then a sequence length for the hash table. This K parameter chooses sort of the maximum length of that. So it's a little bit of an arbitrary guessing game here. Usually I've seen somewhere between 21 to 31 is a good place to be for short read RNA sequencing, around 75 to 100 base pairs. If you have like 200 base pairs, I would go to like 31. It also sort of, when it's computing because it has to compute ties between different sequences, it always has to be an odd number, and it can be as low as 11, I think. So most of the time, this is not going to make too much of a difference in quantification, especially if you have sufficiently long long read lengths for your FASTQ file. So for us, 21 to 21 is fine. We could have gone up to 31, but either way, it doesn't make a huge difference here. So everyone have been able to get this to run?
Yes.
Okay. Let's take a look at what it generated. So it generated this folder that I've called human HG38 underscore index underscore March 2nd, 2023. I'm going to CD into that. And basically what you see here is a bunch of flatter files, right? You see it does a very similar thing to what Star did, where it took our two reference files that are very big files and it chopped it up into a bunch of smaller files. And here we have 3, 6, 9, 12, 15 files. They're mainly these .bin files that, let's see if we can read them. Hello. All right, so my terminal is not responding. There we are. Okay, this is okay. Okay, that's fine. Let's just my God, it's being slow. Sorry about this, guys. So what you can see that is that most of these are bin or JSON files, right? These bin files are binary files that tell you things about positions in the cDNA FASTA, annotation for the cDNA FASTAs, all that stuff that's going to be relevant to us when we're mapping. And the JSON files are sort of tab delimited files that store these hashes that we need to run against, I guess. Okay, so the next step here is to quantify our reads. So now what we're going to do is change our directory to our data. All right, I'm going to end this terminal. I'm going to open up another terminal. Okay, so while I reconnect to the terminal, let's chat about what this next chunk of code does. So I'm changing the directory back into the data, the data folder that we have. I'm also generating another variable here that points us to the path for this reference transcriptome that we generated. It's this human HE38 index 030223 that's inside my reference. And I'm going to run quantification for all of my FASTQ files through this for loop, which is going to be a little bit different. So this for loop is looping through any pattern that matches the asterisk here, such that that asterisk is followed by a .fastq.gz. So let's take a look at what that sort of means for us. So if you go into your data file, you're going to have a bunch of FASTQ files. For us, it's kd1.fastq, kd2.fastq.gz, sc1.fastq, sc2. There's also the trimmed kd1. We could run that as well. That's fine. But that's going so for any sort of loop in Unix, you have to follow the loop with a do command. This is all stuff that we cover in the Unix workshops. And essentially, for each iteration, we have a set of commands that we're telling the sort of command line to run. First of which is creating a prefix. What this does is it takes this asterisk part of the pattern and saves it to something called base. So this is sort of the base prefix of our file names. And then we're going to run Salmon. We're going to call Salmon with this variable. We're going to say we're quantifying. We say that our index folder is saved in this variable name. This -L says that we're automatically trying to figure out what the library type is. -R is going to tell us what the file is. -P is how many threads to run on. We're going to run on thread here. -O tells us what to name our output files.
Find out, tell us what the name.
The sort of -O is going to tell us. Sorry, my Siri is acting up now. Okay. -O is going to tell us what the output is going to be saved as. It's going to be something that looks like the base name that we saved, the prefix that we saved into base. And then we have some other options. We have our sequence bias correction. We're going to add in GC bias correction. We're going to tell it to use this variational Bayes optimizer, just because that's available to us, just looking at different parameters. And lastly, we'll tell it to go back and validate the different mappings it uses to run the quantification. So are there any questions here?
Nope.
Okay, so let's try to give it a whirl then. I'm gonna paste and enter. So what this is going to do is go through every single one of the FASTQ files that we have, and it's going to quantify expression for that FASTQ, and save it in the same folder that we have it in. Okay, so let's take a little
some of the techniques. So for those smaller experiments, you use some of the techniques we talked about yesterday.
Yeah, I mean, so it doesn't. So that's the other thing. So I personally continue to use Salmon even for smaller experiments, just
Okay.
because at the end of the day we're using similar annotations. So the transcripts that we quantify, or the exons that we quantify, are being represented in both similarly in both settings. But I'm at the point where now I'm working with long reads and trying to assemble or trying to identify novel exons or alternative exons or novel transcripts, and that's where an aligner is extremely important, even for quantification, because we want to be able to move beyond the annotations.
Thank you.
there's also literature. We'll take a look at this, and once we've quantified, and we can talk about some of the literature. But there's not really a huge difference in quantification, what using Salmon and using HT-seq or using RSEM. And since there isn't a huge difference, oftentimes once you have hundreds of samples, that's where it's just obvious to be using something like Salmon. Okay, which number run am I on? 1, 2, 3. Okay, so I'm around halfway done, I would say.
So sorry, Arjun, are you running the index, or you're actually the next step beyond where you're doing the?
I'm running quantification right now.
Okay.
Yeah,
I'm behind, but I got the index, so that's
perfect.
good.
That's great. And as you can see, we're getting mapping rates of around 90% here. So, like I mentioned from yesterday, like right now on the screen, it tells you the mapping rate. So for this specific sample, 89% mapped. That's generally in that range that I mentioned. It's 75 to 90% of reads being mapped to the transcriptome through a reference transcriptome.
So when I do the for loop,
Yeah,
it's giving me the it says the following errors were detected within the read files file *.fastq.gz does not appear to exist.
so are you in the folder where all your FASTQ files are?
Oh, let me check. Sorry, I feel like this is a recurring thing for me. Okay. The FASTQ files should be in data, right? Or in reference.
In data, yeah. I mean, it's up to you. Like it depends on where you kept them, right?
Yeah. Okay, let me see. So yeah, I'm in. Okay, so earlier I was in reference. I now switched to data because I was in the wrong place.
Mm-hmm.
And now it says the index version file human. Oh, is it because we're doing HG38 and not 19?
I mean, again, it depends on what you named it as, right? This is all like
Yeah.
you don't have to do it exactly the way I did, but you have to do it the way that you did, right?
Yeah, that's true. Okay.
Yeah.
Yeah, it keeps saying it's not invoked properly. I'm trying to figure it out.
You want to share your screen?
Yes, sorry.
Yeah, no, this is what this is for. Um,
Oh, hold on. I think I also realized um no, never mind. I thought I saw something, but I guess I'm not anything.
what is it? What's the line of code you're in here? Can you go up?
Yeah.
Let's see. Can you press the up button on your keyboard? All right.
Yes.
Oh yeah, so yeah, now you're not linked to like you have to. So this line that I had where it says index folder equals.
Yes. Uh-huh.
So you have to like put the full path to the index, which should be in the reference folder, right?
Yes.
So
Oh,
okay.
yes. Thank you so much. Sorry.
I think I'm stuck in the same place. Arjun, can you show your text file?
Yeah,
Where
1 second.
show that.
There you go.
All right. Get on my reading glasses here because it's small.
Oh, I can make that bigger. 1 second.
Cool. Okay, so it starts with the CD and to the index folder. So that whole stretch needs to be in there.
Yeah.
Folder equals.
Yeah. So I mean, this is again specific to me, but I've sort of
Right.
but change into the data folder. Make sure that the index folder is like saved as a variable, the full path to it. And
Okay.
this makes the code be a lot neater, at least for me.
Okay. Human HG index. Thank you.
Mhm.
All right. Data.
Okay. Slowly getting there. Oh, that's not it.
Arjun,
Yeah, what's up?
can I also share my screen?
Yeah, go ahead. Let me stop share.
Here, I'm not sure what's the problem. Is not working.
Did you define salmon properly?
Yes, I think.
So type $salmon space just in your terminal. Type $salmon in your terminal.
Okay.
Type $salmon.
Oh, it's actually, I don't know why it's too slow.
It's too slow. Oh.
I press, but it's not give any response.
Oh, that was happening with me as well. So
Right now it's not working.
okay, so
Nope. Okay, actually, when I check the path, it's look correct, but do you think is there any problem with the Salmon location?
yeah, so like there's two things. So what that basically means is that you didn't define the salmon variable properly.
Actually, we have a two paths here, right? One of them is the sorry, where is it? Actually, my all data is in data folder, and I defined the index folder.
Okay.
Index folder as in the data folder, human, and you can see here. Is it here?
Okay. But then the error is about the salmon executable. So what did you save as the salmon variable?
What's the mean of that?
Go back to your code, the sort of notepad that you have open.
Actually, I use this pad.
Yeah. Go up.
You wanna run me again, or
No, no,
you
no.
want?
Scroll up. Scroll up. No, no, no. In your notepad, scroll up.
Okay.
So with that ran properly, right? With
Yeah, the first one is run properly.
okay, so how that's weird. Oh, no, what? Okay, do you mind if I take control of your screen?
Yeah.
Okay. Okay.
You take it.
Yeah, I do.
Okay.
Okay. Oh, you're back in your login. Okay, QRSHLHRG equals to hope I tapped all that properly.
I close 2 times and it's same. It's the first is same, not change.
So the issue that seems to be is that since you closed and opened again, yes,
Yeah. Yeah.
this variable wasn't like you have to run this again.
Oh, I see.
So and then instead, so this will and instead of this, you're gonna have to do index folder, okay, and make sure that these letters here are proper, right?
Mhm.
So that should work. What I typed there.
Okay.
So try that and see if it works.
Okay. Okay, I will try it.
Okay, so let me now share my screen. All right. Okay, so now I'm done. So let's take a sort of minute and think about what we did. We quantified, for me, 5 files in sort of 20 minutes. And we did it serially with one thread. So now the idea here is that you can essentially run this with multiple threads and parallelize parts of this loop. So it'll be way quicker than star can ever hope to be. So this is part of the reason why we use Salmon to quantify expression when we have like hundreds of samples. In terms of the output that's generated here, for every FASTQ file, it's generated a output folder, kd1.salmon, kd2.salmon, sc2.salmon, and sc1.salmon here. Let's take a look inside of those files. So it's lagging a little bit. That's fine. I'm gonna print the contents here. Okay, so this file has 3 folders: an auxiliary information folder, library parameters, and logs. So logs are going to contain information about the salmon run. Library parameters are about the library, how many reads are mapped, where the reference transcriptome index is. Auxiliary information tells us things about the FASTQ files themselves. And there's some JSON files that sort of contain all that information in a sort of tab delimited, easy to read format. And this last folder, all the way at the end, quant.sf, is exactly where we keep the quantifications. So as you can see, I did a header on this. It's quantified transcript level expression. ENST IDs tells you the length of that transcript, tells you the effective length, which is sort of a relative normalization of the length, gives you a transcripts per million reading in the fourth column, and then the raw number of reads in the last column. And we have this for all 4 of our files. We have a quant.sf in the outputted folder. Any questions about this?
Nope.
Great. So while that runs for everyone, or while that finishes running for everyone, let's take a look at can everyone see the slides?
Yep.
Yes.
Okay. So one last bit for today. We're going to take these quant.sf files that are contained in these folders, and we're going to import them into R. And we're going to be using it's a package, but I like to think of more of as an infrastructure that's called TXI meta. So the idea here, right, is we've done the blue parts of this blue part of the pipeline. We have an RNA-seq FASTQ files, raw. We've quantified them using Salmon. We have our output right now. We have our output with lots of different associated auxiliary information. We also have our sources for our reference transcriptome, which is in our FASTA assembly. We've indexed it, and at every single sort of points of generating different intermediate files or downloading reference files, there's something called a checksum. And these checksums can be linked together in a table that can then sort of give you a linked what we call provenance. And what these linked provenances do is make it really easy to see where all the shared information lies across your different file systems. And using this one command in R, in the TXI meta package, the TXI meta function in that package can take all of the data that we have in these quant.sf files for all of our RNA sequencing quantifications, and it'll read that in and save it as a very tidy object in R called a summarized experiment. And then that summarized experiment integrates neatly with different kinds of differential expression software methods or software for packages for normalizing data, removing batch effects, visualization, network analysis, lots of different kinds of things that you can do post quantification. So this sort of object in R is all you'll need to run these analyses on gene and or transcript level expression. Any questions about this?
So this is the output data that will then be used in all downstream visualizations and
Yes, exactly.
analysis. If we have a second, if you wouldn't mind looking at, I think I'm having the same problem that Mustafa was having on.
Yeah. Let me go ahead and share your screen. Yeah, there we go. Let's see.
So this is correct to where the index is.
Yeah. So the only thing you'll have to change is in that line that says $salmon quant.
Yep.
So you're saying what that says is look in the current directory for that folder. Instead, you want to give it the full path. So just $index_folder. Or just copy the entire path. Yeah.
So $.
So instead of after -i, that -i is asking for the index folder. And so instead of the name of the folder, which is in a different directory, you can give it the variable that contains the path that you defined earlier up.
I'm not seeing it.
So it's right there, the index underscore folder.
Index underscore folder. Why am I?
It's right above the first line before the for loop.
There. Okay. Yeah.
Yeah. So so you can just use that variable instead of the name. So where you run Salmon down there, you can substitute $index folder instead of human underscore HE19. There you go.
And then
That should
base. Okay.
that should work.
Thank you.
Maybe not. Oh, never mind. Oh, never mind. It's it might be that in your so are all of your fast that's because you unzipped everything, right? So
yeah.
should be fast queue, yeah.
Fast queue.
Yeah.
Fast queue. Okay.
So in your for loop, just
here.
delete the .gz. And the same thing in that base name part as well. Yeah.
I'm glad you remember that I did that.
Huh. And why is it? Oh, and now your quotations. So you're gonna have to.
I thought I'd replace. Oh, okay. I know what I'm.
Yeah.
Yep. So Control C. Go up to.
Yeah. So if you Control C again. If you press up in the terminal.
Press Alt in the terminal.
The up arrow key.
Up arrow. Okay.
Okay, so now you can just that's the whole command, just not written in like multiple lines. You can just go back to the quotes and fix those.
Is it just this one here?
It's both of them, I think. You just have to replace with with that.
Oh, there it is. Okay.
Yeah.
Okay. And then I can just hit return.
Yep, that should work. Oh, and then done.
Dot.
Done.
Done.
Yeah, that should be good. There we go.
Okay.
All right.
Oh, thank you.
Yeah. So it should take about 10-15 minutes to run, but
okay. So I'll catch up eventually. I'll just watch you guys do the rest, and we're moving on to the R.
Yeah.
Okay.
Everyone else? Anyone else having trouble running the quantification step?
No,
I'm still
but
in trouble, but I don't want to keep you.
Oh no. I mean, we have lots of time left, so
yeah, it's still going.
still going, or is there an error?
No, it's just still like around 50%.
Okay, all right, that's fine. Anyone having errors?
Did we have to load fast queue? Mine's just saying that my file doesn't exist, but I think I might have pointed in the wrong direction.
Do you want to share your screen?
Yeah, one moment.
Oh, I am sharing my screen, so I should probably stop my share. There we go.
So currently trying to scroll up. Oh, that was too far. Okay. So I got fastq does not appear to exist.
You're in the reference directory.
Oh, that's definitely it. CD, data was data where it was, right?
Yeah, yeah. Now, given that you linked to the index that's in your reference subdirectory, it should run. Which you haven't, so be careful.
Oh, whoopsies. Hold on. All right, so I just need to say what index is.
Yeah, so just define another variable for that.
Yeah, get it in the index equals section. Sorry, give me a second. Index. I can get it from here. Thank you.
Great. Anyone else with errors?
So my error is that it keeps saying it says bash and then it gives me the the pathway, right? And then it says is a directory instead of like executing.
All right, share your screen. Let's take a look.
Okay.
Yes.
Okay, so here I like control L's because I was getting so overwhelmed, but I'll scroll up. I'll scroll back up. So like if you look here, if you can see my cursor, like the third line, this is what kept happening. And so like I started from the very beginning in terms
Yeah,
of
you just have to add a a slash salmon to that, right?
oh my God, seriously?
Yeah.
After software and before the salmon thing.
Yeah. So after bin, after bin, you just have to add /bin/salmon. So you're pointing it to the directory where the executable sits,
Index.
but now you have to give it to the you have to give the path to the you have to specify the path to all the way to the executable.
Okay, so sorry. Okay, so let's say I just want to start blank, right? Because I think it's easier for me to visualize. Do I do this index underscore folder first, or I have to do the salmon thing you were talking about first?
It doesn't matter. So these are just defining your variables, right? You're
Okay.
saying that in my index folder, this is the path. Now you're saying the salmon executable, here's the path. So you can do it in either order, but you have to define those both before you run the for loop.
Okay, so here, let me rewrite the salmon portion of it so that way it's listed. So this is what it should look like.
Yeah, but then /salmon.
At the end as well.
Yeah.
So salmon after software and salmon also
No,
after.
not after software. So
Oh, okay.
yeah. So remember, like these are the way that these like each slash is like if it starts with a slash, right? It's a
Yeah.
directory.
Okay. So here, let me try this one more time. I'm sorry. It's hard
Yeah.
to visualize this. So there, okay.
So now just to test that out, type in $salmon -H, enter. There you go.
Okay.
Salmon's defined properly. So
So
now you can
find properly, and
so
then
now you have to be in your data folder or whatever folder has your FASTQ files,
okay.
and then now you can run that for loop.
Okay. And do I need to redo the index folder assignment, or it will still remember it?
It'll remember it, yeah.
Okay, so let me try doing this again. And I've changed the salmon quant -I to $index folder, right? Because I remember you saying that earlier.
Yeah.
Okay, so I hit enter.
Error file fastq. It should be fastq.gz, right?
Oh, yes. Okay, so Let's try that again.
There we go.
Ah, success. Thank you so much.
Yeah. All right. Anyone else?
I just miss where you find the human HG33 index name. Because in the exercises document is HG19, but you check somewhere and put here the 38 index dot 0302223.
Yeah.
But I just copied your text editor screen, but I miss where you find it.
Yeah. So again, this is all dependent on how you're saving things, right? So this let me share my screen. All right. So the folder that I named it as was this, and this was in the reference folder. So that's why I saved my index folder path as in reference.
Oh,
And
is it not specific? So when we write with the HG38 or blah blah is not matter, is defined by ourselves.
Yeah, it's what you
okay.
saved as. Yeah.
Okay. I just think that is there some kind of specific things, so I asked. Okay, thank you.
Arjun, my run is finished, and I'm just trying to check whether I got what I want, but I'm not sure whether it's saved into the data folder.
It should be, yeah. So what you should look for are folders in there that are like kd1.salmon or kd2.salmon. That's what you should be looking for. So right now, for me at least, I'm lagging a little bit, but I'm in a folder called kd1.salmon, and it has these files inside of it. This auxiliary info folder, library parameters and logs folders, two files that end in .json, and one quant.sf. And you should have one for every single one of the FASTQ files that you have.
So this is kd1.salmon should be in data folder. I don't think I have that, so.
It should be in whatever folder you ran the salmon run in.
Okay, thought that was data.
I thought so too. Were there any error messages that were printed?
Yeah, I could share screen and show you what I got if you want.
Yeah, that'd be good. Do that. Oh, I think the other issue is that it's that's weird. It's like
Could
the
it be?
asterisk also. So press up again a couple of times back to that for loop that you ran. So go back to the for loop, the for part and the asterisk, and retype that asterisk in there.
Is there a smart asterisk that's getting screwed up?
It could be that the sort of RTF file that you're using, RTF, that asterisk looks different and registers as different. And then, yeah, press enter again. And then done, yeah. Okay, that's it's the same. Okay, press end it again. And then press up again.
So I have to change the quotes. And
Yep. Yeah, the quotes there.
I've changed the preferences on my text editor to not have smart quotes, so I might be making progress. Was there anything else? The asterisks, the fast Q?
I think we're good, yeah. Scroll up a little bit if you can. Or just with your mouse, yeah. KD1.fastQ. Okay, yeah, well, you should. Oh, output is the base name. That's an issue.
Written to base name.
Yeah, that's the problem. Okay, so
Control C.
yeah, I mean, yeah, so can you go back to your code for a second? Let's see. It's very weird. Yeah, I'm not quite sure what's going on here.
Okay.
So what I would do just to make sure that you have a single run done here.
Yeah.
So for this like salmon quant right here, that section,
Yeah.
instead of $fastq or $FQ, just write out KD1.fastq. All the way down, like where you run salmon itself.
Here.
Yeah, just change that into KD1 dot fastq and no $. And then for base, just change that to KD1 as well. No $sign as well. And then just only run that part, the one that just before, you don't need the done. Just that part that you've yeah, right there. Just run that part and then you'll just have KD1
Okay.
done, at least. Let's take a look if that runs properly. Okay, at least we're writing to. Okay, cool. We should be good to go.
Okay. Yeah. All right. So somehow I'm just not.
Yeah,
Right.
it's just that one line of code that's creating the variable name base.
Yeah.
It's not sort of recognizing the command base name.
Yeah. Try rewriting it.
You can try also double quotes instead of a single quote and see if that helps with the smart quotes issue there. But at this point, I think just having it done for one sample is sufficient.
Sure.
Yeah.
Yeah. All right. I'll do that.
Okay. All right. So so now what we're going to do is I'm going to be opening up a RStudio shell. You can also use a text editor if you do. But if you're going to be doing bioinformatics, I personally would always have I would have RStudio on your laptop. So everyone have RStudio or would you like to sort of so RStudio is a free it's free from Posit, which is sort of what R has rebranded itself to. And if you look at products, there's the RStudio IDE. integrated development environment, and you can download this RStudio desktop that'll execute R and Python code directly from your editor. So it's a very good way to just run the things you need to run. Most of you are going to be running commands in R and Python, and so this will be a good thing to have on your laptop. Just download this, and before that, so we're not going to be using R on our local machines, but if you do want to run this on your local machine prior to installing RStudio, you'll have to download R itself. So you can download R from r-project.org, and it's available for Windows or for Macs. Oops. So you just search it on Google, you download the package here, install that first, and then go to posit.co and download the RStudio desktop. Regardless, I'm using it mainly as a text editor right now, so it shouldn't matter if you are using the text editor that you have currently. But what I'll be doing is sort of manipulating the code in RStudio and then just pasting it into our terminal. So I'm going to cd into the data folder where I have, or wherever you have your .salmon folders, the output from Salmon. And the first thing we're going to be doing is installing TXI meta from Bioconductor. So it's this package here. So we're going to, like we did yesterday, we're going to load up, not add, load. Alrighty, come on. All right, we're gonna load this 4.2.2 -bio module for R, and then we're gonna type capital R enter to enter into an R session. And then from there, we're going to install TXI meta from Bioconductor. Okay, so while that installs, let's take a look at what I've done here. So now I'm going to be loading up, bringing that installed package TXI meta into this current R session. And now I'm defining a data frame here. And
So just a quick question, can I go back to the installation? I think I missed the very beginning.
Just this, that's all. So I typed in module load r/4.2.2 -bio, and then just R, then enter. Okay. And then when I got into R, I installed TXI meta. Okay.
Thanks.
All right. So now I'm going to Ooh, this is very slow. So now I'm going to be loading up the library, the package TXI meta that's in that I just installed. And I'm going to define a data frame. So in R, a data frame is essentially like a spreadsheet, a table. And if you use this function data frame, data.frame, you can give it multiple options that essentially define each column. So here, what I've done is this data frame is going to have a first column that's named files. And as you can see, these are the file paths to the quant.sf files for KD1, KD2, SC1, and SC2. So currently I'm in the data folder. The data folder has 4 subfolders, KD1.salmon to SC2.salmon, and each of those subfolders contains a quant.sf file. And I'm just writing those paths out in a single and saving that to the first column of this data frame. Okay. The second column now is names. I'm giving each file the proper name. I'm saying the first one is KD1, the second is KD2, the third is SC1, and the fourth is SC2. And then I'm giving them conditions. I'm saying that the first 2, the KD samples, are knockdown samples, and the SC samples are scramble conditioned. So a knockdown and controls. And then I'm making sure that the strings are not vectors. And at the end, what this call data looks like is, let me print out what this call data looks like. It's just going to be a table with 3 columns, files, names, and condition that exactly look like what we defined here. This last line of code here converts these strings into factors, because that's going to help us. It'll help us with downstream sort of things. So I typed in call data, pressed enter, and the output here is just exactly what I want to see. A column named files with the 4 paths, a column named names with the 4 names, and a column named condition with the conditions for each of these samples. Is this clear to everyone right now?
Yep.
All right. Now we're going to run TXI meta. So based off of this data frame, it's going to require a column with files and a column with names, and that's all it needs. And you can also start tacking on other kinds of variables as well. So say you have like a population setting here, and you have like a bunch of different names. You have like a condition for like disease status, but then you have like other variables you want to control for in your experiment, like age and sex and sort of hospital visit and things like that. You can tack that onto this call data, and it's going to integrate it seamlessly in this one line of code. So I'm going to press enter now, and it's now starting to import the quantifications. Just reading in the 4 files. It found a matching transcriptome because of the checksums that we generated when we ran this. It found a matching GTF file from Ensemble, and now it's downloading that and keeping it in this sort of like safe directory that's specific to this sort of reference transcriptome database that TXI meta pulls from.
Arjun, how did it know where to look for the reference file? Is that something that's encoded in the checksums that come with the.
Exactly, yeah. So those files that generate inside of the sort of kd1.salmon folder, et cetera, it tells from there's information there about where the reference transcriptome from Salmon is. And from that reference transcriptome, it'll know where the source of the annotations are. And from there, it knows that it's from Ensemble, it's release 109, it's this build of the genome 38, and that's why it's specifically downloading that GTF file here.
Great, so that's nothing we need to specify.
No. So to process the genes, process the transcripts, looked at exons and chromosomes and metadata.
I have a quick question. So in the cold data, I listed like all 4 files, like the KD1 salmon / quant SF, SC1, KD2, SC2. But when I put cold data into the Linux, into the command line, it only shows 2 rows instead of 4. Is that like, is there a typo somewhere that it's not reading the other 2?
Yeah, do you want to share your screen?
Sure. Okay, so here, I guess I'll share my RStudio first. So this is what I had written down.
Yeah. Oh, yeah, you forgot commas.
Oh,
8 and 9.
on lines 8 and 9.
Yeah.
Oh, I see now. My goodness. Okay. So this and there.
And then
That's-
also commas after KD1 and SC1.
Like here.
Yeah.
Okay. Oh,
No.
that's
Yeah,
okay.
after quotes. Yeah. There you go. That should work.
Got it. Okay. So then I have to, I'll reassign this in the command line, right?
Exactly. Yeah.
Okay, so let me just make sure. So if I do that, and then I do call data.
Mm-hmm.
Ah, okay.
Cool.
Thank you.
Of course. Okay. So the next thing we need is to install this package, Summarized Experiments. Summarized Experiment is, again, more of an infrastructure than, oh, it's already in there, so we're good. Never mind. Summarized Experiment is more of a infrastructure than a package because it sort of defines this data object in R. So let's load that up. I'm going to load up this library. It's all lagging a little bit, so my apologies. Question?
Your screen's not shared right now.
Yeah, can you share your screen?
That is very important. So all I did was type in this command here, library summarize experiment, the one that I've highlighted. And okay, so now I've loaded up the summarize experiment. Let's take a look at what this SE object is that we saved in. So what we see here is this is a class in R, a class of data called the ranged summarized experiment. We have our dimensions here, 181691, which is the number of transcripts that we have. We have 4 columns. And the metadata that we have is information that comes directly from the Salmon output. We have 3 assays here, counts, raw counts, the normalized abundances, and then the gene lengths. And our rows are these ENST values. So these are transcripts. So let me try. So if we sort of, let me see if I can pull up a summarized experiment R. images. Cool. So the way that a summarized experiment works in R is that there's a set of assays here. For us are these three assays of count, abundance, and length. That's what the main stuff is. Along the columns are our samples, and there's column data that's associated with that. And we have 4 columns, KD1, KD2, SC1, SC2. We have associated column data, names, and conditions. So this tells us that KD1 and KD2 they're forever linked with being a knockdown condition. SC1 and SC2 are linked with being a scramble condition. Similarly, we have row data as well, which tells us information about the transcripts. We have transcript ID, the external name, all the way down to gene ID and other kinds of information here. So we can extract all of that by using the function row data, row capital D data, and then put SE inside of that. And once it reads out, what you'll see is it's a data frame that contains all of this information. So essentially it's a series of linked data frames and matrices that make sure that all of the information that you have about samples and about the features, the transcripts or the genes, they're in the same place. And then you can start adding metadata to it. You can add models that you're interested in. You can add information about the libraries, things that are important for the analysis, but aren't specifically linked to the samples or to the features. Oops. Ooh, this is all right. See if there's a row. Okay, so here's the row data. So it's a data frame with 181,000 rows and 7 columns, transcript ID, transcript external name, transcript biotype, the start and end of the of this transcript, the gene that it belongs to, and then the transcript ensemble ID here. Similarly, if we look at the call data, it's 4 rows, 2 columns, names, and condition. Exactly what we gave it in the call data data frame that we generated to read in the data. And then lastly, we can do access the counts from the assays by just accessing it as a list. And again, we're looking at transcripts, so we have lots of sparsity here, but it gives us the matrix of counts, transcript counts that we have quantified using Salmon. Okay, one last bit here. One last bit here. The last bit here is to take our transcript counts and summarize them to the gene level. So the idea here is that gene expression is just the sum of the transcripts or sum of the isoforms that it had, some of its isoforms. And now we can just sum those up and summarize in another neat and tidy table and save it as GSE, gene level summarize experiment, and use the summarize to gene function that's going to help us do this. And once my terminal responds, I can type this in. Okay, come on. Okay, are people able to run this TXI meta and run the last bit of code as well?
I don't think I've got the R code quite right, but because I had to change it because I only had the knockdown and not
Yeah.
the sound. So I won't take a quick look at that, but it might be better to help somebody who has all the data first.
My salmon quantifications only just finished like a minute ago, so I'm trying to
Okay.
catch up from BioC Manager, but I should be okay.
Okay. So it really shouldn't be that much different for you, Tim. So you can share your screen and we can take a look while my terminal decides to.
So I don't have our studio, so I was trying to just do it in the text editor, which may also create problems. I've got our studio, but it's not opening because it's an old version. So I went through and eliminated from the version that you had in the exercises, which had 2 levels, KD1 and SC1.
Yeah.
And so I just eliminated those and the comma and closed the parentheses.
It should work, yeah.
So let me show you that. So there, data frame equals KD1 closed.
Yeah, that should work.
Names equals KD1, comma, condition equals knockdown, comma 1. Is that correct?
Yeah.
Okay. Strings as factors false. Looks like I'm missing a parentheses here. I don't know.
I don't think so. It should be fine.
Okay. And then cold data condition, cold data.
Yeah, I think you're missing a parentheses there at the end of that.
Here?
Yeah. Okay. And now that should run properly.
Everything from library.
Yeah.
TXI meta. Okay. All right.
Well, let's see.
Parentheses somewhere.
Is it the quotes again?
Mother fudger. Could be. Yeah.
Yeah, I think it's the quotes for the condition, like the second time, the call data $ condition, that line.
Mm-hmm.
Those quotes are. It keeps, yeah.
Keeps doing the same thing. I've got smart quotes turned off here. Text replacement. Got this as a plain text.
Yeah. It really wants you to use those quotes.
It really does. Well, this is my personal hang up. I'll have to find a text.
Yeah. So like ideally you don't have to have the second line. So you can go ahead and run the TXI meta function to the next line there. SE assigned to yeah, you can go ahead and run that. It should be fine. There we go. And all that, yeah, is yes, yeah. Yes, to that as well. There you go. It's downloading your
Okay.
so it's using or it's generating a temporary cache, and it's going to start downloading the GTF after this.
So the stuff that we skipped over is just defining a data matrix, but since I only have one instance, I don't need it quite so much.
Yeah, not just that. It's what it's doing is making sure that knockdown and scramble are no longer characters, and instead they're factors. And in R, if you define something as a factor and then run a regression, it's going to treat it as like as a categorical variable.
Okay.
With like with a certain reference, as opposed to a character variable that kind of like craps out and turns into numbers, and then it's not really interpreted as the same way. Okay, so my terminal is now not really cooperating. Has someone been able to run SD that TXI meta run all the way through?
I think I have.
Do you mind sharing your screen again, and we can try to. So here, if you run the next code, the GSC, or the next couple of lines here, I think you might have to load up the library as well. So what this is doing is grouping the transcripts that belong to the same gene together, and then this is quantifying the gene level expression. So it's summarizing abundances, counts, and length. So Tim, if you type in GSE enter now, what you'll see now is the dimensions have really decreased. So this is now 37,000 features. And if you look at the row data now, it's starting with gene ID. And the row names are ENSG ensemble gene IDs instead of ensemble transcript IDs. So now this is effectively the same sort of output that we ended up with yesterday at the end of our work, or end of our session. We have a gene count matrix for however many samples that you quantified, with information about the raw counts, normalized raw counts, normalized counts, normalized abundances, relative abundance, and length of the gene, all in a very tidy, ready to use format that integrates with different kinds of packages in R.
Cool.
So that's all I have for Salmon. So if there's any questions, we can take another sort of like 10-15 minutes so that everyone is able to run through this and have been able to do this on their own. But that's all I have. I'm gonna share my screen and have the QR code up, so that everyone has that. But I'll stay on for any questions. But aside from that, that's all I have for the workshop. Quiz and homework due next end of next Friday, and I'm happy to answer any questions over email as well.
Thank you very much for teaching us.
Thanks,
Yeah,
Arjun.
Yeah, my pleasure.
Thank you, Arjun.
Thank you.
So just a quick question: how do you get out of the R environment in the terminal? Like
Control Z.
Control Z. Okay.
Yeah.
Thank you.
Arjun, has R created any additional folders here? Is any of that, how is that information saved?
Yeah, that's probably a question I should have answered. So you can sort of either save your entire R environment as something called an R data. So you can do save.image. So if you just so this right here, save.image, and you can give it a name inside of these brackets here.
Mhm.
That'll save everything that you've created in R, and you can load that up using load. The other way, you can just only save, say, that SE object, I guess, is you can either do so save.image W5a txi meta.r data, or I'm sharing my screen, yeah, or save.rds, save rds se transcript level expression and W5a dot RDS. And what this will do is this will save everything that's been generated into this R data file and save it exactly where you are. And what this does is only saves this object as a file that's specifically is specific to R itself. And you can load that up with read RDS. And the other way, of course, is I would highly discourage this. But if you only sort of need the count matrix as an Excel or something, you can always save or write.csv that as a CSV file. But I would prefer to do it as an RDS, keep the objects as different RDSs, or so that when you're analyzing it in R, you can just open that up and start from there. Does that help?
Yeah, thanks.
All right.
Actually, my run is still running. Should I wait for it or?
It's up to you. It's up to you.
Because you know the terminal is
Slow, right? Yeah.
very slow right now, so I'm not sure. Start again later or?
Yeah. So all this is recorded, and the last term's recordings are also available. So if you don't want to wait until it finishes here, you're totally free to go. But I can be here until 4:30. If you run into errors or anything.
Okay, thank you.
Yeah.
Hi, Arjun.
Yeah.
Can I share just my screen?
Yeah. 1 sec. Yeah. Go ahead.
Uh,
Let me stop my.
yeah.
All right.
Can you see my screen now?
Yes, I can.
At the very beginning, I want to do this assignments, and I found that it can't find there is an error here. But I do check that this JSON file it would.
Did you name it properly? So did you name it HG19 or did you name it something else?
The same thing. You can go back to see the folder. It was. It's here. So this is a folder, and the file is here.
Oh, that's odd. Did you give it the right path all the way through? Can you go down?
Yeah. The path is here.
Oh, it's software. So that's the thing, right? It's reference, right? Not software.
Oh yeah. Oh, see, software. Got it.
Yeah, that should be that should fix it.
Yeah. Okay. Thank you.
Yeah, of course.
Try it out. Okay. Mhm. Mhm. Oh, wow. Mhm.
I'm heading off, Arjun. Thank you very much
Yeah,
for
thank you so
assistance.
much. Yeah, appreciate it.
Bye.
Bye.
Yeah, thank you
All right, thank you,
for
Mustafa.
joining us. Bye.
Yeah, definitely. Okay, bye.